Okay, everyone, Dr. Phil here. So I want to sort of start with some of these PowerPoints. I have some of you for lab. We met Tuesday. Some of you may have Dr. Bailey, and some of you may have taken lab already, or you're taking it somewhere else. So this chapter is really going to talk about lab identification. So how are some of the ways we can identify what something might be? So Tuesday, I gave all of you, if you're in lab, some agar plates, agar, either a nutrient plate or a tryptic soy agar plate. So these are just general nutrition media that most things will grow on. So your homework was to kind of swab something, and we talked about, whether it was the bottom of your book bag or purse, or your keys, cell phone, keyboard, kitchen sponge, a flush handle on your toilet. We talked a little bit about like the Sonicare. If you take the brush off, like there's always stuff growing underneath the where it attaches. Just kind of get in your head, like where would you swab? Or if you had a, let's say you had a wound or an infection, you could swab it. We would grow it, and then we do a gram stain, and we do some kind of selective culture on it to see what it might be, or what are some ways we can identify it. So I have a bunch of stuff here around me. If I can get to it, I don't know if it'll work with the camera, but basically we want to sorry, identify. So isolation. So we're going to just kind of get the sample, isolate it. We're going to grow it. We want to streak plate it. So we have a pure colony with no contaminants in there. We're going to incubate it so it's the right temperature, usually 37 degrees Celsius. It's kind of like body temperature. We'll incubate it either overnight or a day or two, depending on if it's slow growing or fast growing, or what type of organism it is. Then we want to find a way of isolating it so we can figure out exactly what it is. After we streak plate it, we have different colonial morphology. We would sterile isolation loop. We'd scoop up the colony, and we would be able to plate it and see what the organism is. And please realize that a lot of times if you have an infection or if you're on a surface, 99.999% of the time it's going to be a mixed culture. So which means there's more than one thing on that organism. And same with your patients at some point in time. In grad school, one of our instructors used to always say, doctors, don't worry. Your patient's going to have more than one thing concomitantly. They can have ticks and fleas. So just kind of keep in mind that if you're swabbing a surface or an organism or whatever it is, there's going to be more than one thing on there. It could be viruses, molds, bacteria, gram positive, gram negative. Some of them are innocuous, some of them opportunistic, and we'll talk about all those terms. So we want to isolate that. We want to inspect it. We only have bright field microscopes here. If you were here Tuesday night, we had all brand new microscopes, pretty much state-of-the-art microscopes for doing this, and we'll be using them throughout the semester. Then we want to figure out, well, we've isolated it, we're looking at it, we've stained it. What are some of the things we can determine? So whether it's going to be with differential broth tubes, gram staining, we can check drug sensitivity, which we'll be doing. We could do DNA analysis, which is probably the most accurate and quickest. And what they're going to do now, they're going to analyze it with the little DNA tubes. They come in a row. You kind of swab it, and whatever has the antigen antibody reaction, sometimes there's a way of doing that. Or we just check the DNA. They'll tell us exactly, pretty close, what exactly it is, and then identify it. So if we can identify it, we would hopefully have a way of treating it. What would be the standard of care for treating that specific organism? All right, so we can collect this in a food sample. We check for coliforms in drinking water, which are enteric bacteria, and we could, if we're checking the coliforms or we're checking it for lead or whatever, we do a coliform count to see how fecally contaminated might that water be. And anyone who lives around the Hamburg area, sometimes they close Woodlawn Beach. They'll just close it down because the fecal or the coliform content might be extremely high. We could check urinalysis, or we could check blood. So those are just some of the general things. So just start thinking in your mind, well, where could these organisms be? Where could they be growing? How might they affect us, whether we consume it or if it's systemically in our body growing, or septicemia, if it's growing in our bloodstream, that would be a very bad thing, right? So Tuesday, we looked at the microscope. I gave you guys some handouts. It's in your book. So please figure out the magnification and the resolving power, its ability to show detail. And we saw Van Leeuwenhoek in chapter 1. I kind of showed you that not prehistoric, but that very basic microscope that he designed when he was a linen merchant. And then I said he was picking things off his teeth and out of his wig, and animalcules. He saw moving things in drinking water, because they used to collect rainwater in barrels off of roofs for washing and stuff, just because they didn't have indoor plumbing at the time. So and they didn't even know it was, they didn't know what the cause of disease was. They didn't have the advancements we have now. So figure out the magnification, the ability to enlarge objects. And in lecture, we discussed or in lab, we discussed the ocular and the objective. So most oculars, ocular meaning eye in Latin, is where you're placing the eye to look in. Those are usually 10x or 10 magnification. And then we looked at the objectives in lab, and I said there's your scanning objective, which is really, really short. I think it was red. So that's 4, and they should go to increment. The next one should be 10, 40, and then 100, which is your oil immersion. So total magnification is the ocular times the objective. So that should give you 10 times whatever, 10 times 4, 10 times 10, 10 times 40, or 10 times 100. And I mentioned in lab that some microscopes have a 20x ocular. So you would multiply 20 times 4, 20 times 10, 20 times 40, or 20 times 100 to get your total magnification. The maximum being 2000. And then I had about 6 of you ask me, what does TM mean? And I said total magnification. And they, you guys said, well, should I just write it down? I said, no, I think you should write down 10 times or whatever over and over again, figuring out what your total magnification is. So if you're lab practical or on an exam, they said, what's the total magnification? You would automatically be able to say, okay, it would be ocular times objective, and figure that out. Resolving power is the ability to show detail, and that would be your ability to distinguish theoretically 2 things as separate objects, and that's kind of the key behind the Snellen chart or an eye exam, is your ability to determine the difference between 2 objects. Right. So we have a lot of magnification, but if it doesn't give us detail or it tells us how to separate 2 different things, it's kind of really pointless information, right? Sorry, I guess my allergies are wicked today. All right, so magnification is in most microscopes results from an interaction between visible light waves and the curvature of the lens. Very similar to your eyeglasses. If you have glasses on, the light's coming through, depending on the visual spectrum, the nanometers or whatever it happens to be, it's coming through, and your corrective lenses should be able to refract that light so that it hits your cornea. All right, and then your aqueous humor, and the crystalline lens, vitreous humor in the back, and then the retina. So we want that all to line up so it comes into focus. So if you need some kind of correction, whether you're nearsighted or farsighted, it could be an astigmatism, or your corneal curvature could be too steep or too flat, and we want to go in and correct that so that all visible light hits the retina and it focuses there, so that cranial nerve 2 can bring it through the optic chiasm to the occipital lobe, and you can distinguish that image as whatever it happens to be. So the extent of enlargement is the magnification of that. So there are parts of a microscope. This is an old Lycra. We actually have a few of those here, and we have ones actually older than that were here when I was here in 1993. And those scopes are made by Bosch and Lomb, and they're probably from the 70s, and everyone could laugh at them all they want, but they're literally still working, and very little can go wrong with them other than stripping the gears, but they're literally, what, 30, 50 years old, so they still work. All right. And that's what I learned on, and they're still here. All right. There's very little that can go wrong with them. They're not the most advanced microscope, but unless you're really good at using a microscope, you're probably better off with a more basic model. So a Boreal or Boreal would, or we have the new Lycras here. We'll show you how to use them in lab if you want. The worst thing you can do is sit down and start moving stuff if you have no idea what's going on. In your lab instructor, we'll go over how to do that. I went over it in lab, and we had the camera on so you guys could figure out how I was doing all this. So we were projecting whatever I was doing onto the screen, and you could see what was going on. I think we did strep pneumoniae, and we were actually able to see like the shadow around at the capsule. It was super cool. Right? So just know the parts. We talked about PD, or interpupillary adjustment. So some of you were sharing the scopes, but remember I told you to wipe them all off with alcohol because we don't want, I'll show you guys in case you weren't in lecture. So this would be a Dermadex. Hold on 1 second. So that would be the Dermadex. All right, so that is actually a lash mite. All right, that's the growing on the eyelashes. So we want to make sure that we wipe that off. All right, and please realize if you're wearing like fake eyelashes and stuff, they like to grow in that. So know the parts of the microscope, wipe them all off. Oh, I was gonna say, so if you were sharing the microscope, wipe them off. But you may have to move the oculars because everyone's eyes are a little bit different apart. So there's the nose piece, there's the arm. All right, so there's your objective ones, ocular, eye. Objective is the object you're looking through, okay? So there's your mechanical stage right in here. And that you're gonna be moving up and down. And I said you're gonna bring this into focus. Initially with medial is the coarse adjustment. Then you're gonna fine tune it. Then as you click this into the larger and larger magnification, and just go by size. You're gonna go from shortest to longest. All you should ever have to do is move that fine focus a tiny, tiny bit. You should never have to move the coarse adjustment ever again. Right? So there's your stage condenser, which is going to kind of move that iris to determine the amount of light going up through. And remember when I was in lab, when I was focusing on things, especially with the higher magnification, it was going in, I was dimming the light and altering that iris. So we had a pinpoint of light coming up through because it's very, very tiny. So we'll learn about that in a little during the course of the length of the course. All right. And there's aperture diaphragm control. And like I said, that's really to control the iris. Light intensity would be like a dimmer or rheostat, however you want to remember that. So if we ask you in lab, what is this part? Name it, and then tell me what it does. All right. Myself or Dr. Bailey, depending on who you have. All right, and this is really how it kind of works. There's your light source coming up through the microscope, and there is your hitting your specimen on your slide. And the other thing, remember, luckily thankfully, one person had their slide upside down. So everything came into focus really, really well until you got to oil immersion. Then no matter what you did, it wouldn't come into focus. And I'm like, I said, I bet you the slide is up down. We looked, sure enough, the label was facing down. All right, so you want to make sure, by the way, that your sample is superficial. All right, so it's going to be closer to your eye. If it's flipped over, it won't come into focus with the oil immersion. All right, so there's your objective lens, which you're switching back and forth, coming up through the ocular and then your eye, and then I said it goes in the cornea, aqueous humor, crystalline lens, vitreous humor, the retina, with the retina, optic chiasm. Okay, remember the medial crossover, lateral goes straight through the occipital lobe, and you're going to register that in the brain as an image. Right, but remember I said in lecture, the image is usually, it's going to be inverted, could be upside down, depending on how you're imaging it. And remember I said, if you move that stage away from you, the image is coming towards you. If you move it left, it goes right. So just kind of realize that whatever you're doing, don't be shocked if it's opposite. And remember I said too that as you increase the magnification, all right, to make the object bigger or to get a little bit more detail, your field of view always gets smaller. So you could see a whole bunch, but as you go to the larger magnification, the field of view gets much, much smaller. So the real image is projected to the ocular, where it is magnified again to form the virtual image. All right, so it's not a real image, it's a virtual image. Total magnification is always going to be the objective power times the ocular, but I always like to think ocular and then objective, because you're going to, when you're looking at the microscope, you're always going to say, well, there's the ocular where I'm looking through, then the objective. But it doesn't matter whether you multiply ocular times objective or objective times ocular. In my brain, as small as it is, I think ocular and then it's going to the objective to the object. So I just multiply those two. So use 10 times whatever. For here, to all of our scopes here, and most institutions are 10x, unless you're at a research place, there might be more. But resolution, again, I said, is your ability to distinguish between two objects. So the capacity to distinguish or separate two adjacent objects depends on the wavelength of the light that forms the image along the characteristics of the objective. So this is great. It's magnified, beautiful. All right. But we can't, there's no way to distinguish that. Okay. So quantifying resolution. So how do we quantify the resolution power of the microscope? So wavelength is always the top, and that's in nanometers. So whether it's, the phototropic curve, 400 to 800 nanometers, or we can't visualize ultraviolet light, so we only have between 400 and 750 nanometers. 500, in the middle of the phototropic curve would be, red, orange, yellow, green, blue, violet. So that yellowish green, chartreuse is actually the color, is the center of the phototropic curve. It's the most visible light out there. And I guess they tried, years ago, they used to have the experiment when they wanted to do all the fire trucks and all the trucks in chartreuse, that really yellowish green, because they figured, well, it's the most visible light, they can't possibly miss it. All right, but everyone was just trained to look for the red, so that experiment didn't really work. All right, numerical aperture of the lens ranges from 0.1 to 1.25. So always remember, the shorter the wavelength and the larger the number will provide better resolution. So the smaller number at the top, the bigger number at the bottom, gives you your resolving power. So just trying to think, how can I make that number as small as possible? So, well, the shortest wavelength would be the smallest number, and the numerator, and the denominator, the larger that is, the smaller the number is going to be. Okay. So oil immersion objectives resolution is 0.2. Magnification is between 40 and 2000. 40 would be that 10 times your scanning scope, which is 4. Or I said theoretically, the most you could possibly ever get with a bright field microscope is if you had a scope that had an ocular of 20. I've never used one, but they make them. And the oil immersion, which is 100. So 20 times 100 would be 2000. For our purposes here, it's always going to be 10 times 100, which is the oil immersion. So the most magnification we can get here is 1000. And then this picture, if you were in lab, is on the cabinet with the microscopes. And I asked you guys, what, and I did ask you in the lab, what was the purpose of the oil immersion? All right. And some places I've read, it says it helps with magnification. It really doesn't. I guess theoretically, if you look through a drop of water, the curve of it could magnify things, but for our intent and purposes, the oil immersion is to make a space between the slide. We're not using cover slips in here when we start making our own slides here. Unless we have a really good slide, then we'll put some permamount on it and we'll slap a cover on it and we'll save it. But it's going to seal the slide to the objective. And the oil immersion has about the same density as water. So the refractive rate, so the light will go right up through. If we don't have it, some of the light scatters and things won't be as clear because we want all of the light beams going straight up through. We'll still get some of them coming up, but the more we get, the more the clearer the object is. So these things here are not resolvable. These are resolvable. And in lab, we looked at the cocci, beryllium. This could be theoretically rods, depending on the size of it. It's curviform. So we're not really worried about the shapes for this slide right here, but just try to keep in mind, we did look at some of those bacterial shapes. But this is our ability to resolve these things as separate objects. Okay, so here's a bright field, most widely used. Specimen is darker than the surrounding field, used for live and preserved stained specimens. So that's kind of what we do here. We're not going to be doing anything live per se. Yeah, we really don't have time to look at pond water or something like that in summer. Dark field, brightly illuminated specimens are surrounded by dark field used for live and unstained specimens. So you need contrast against your dark field. So here's your phase contrast. Fluorescent microscope. These are kind of cool. So they're a modified microscope with an ultraviolet radiation source and filter. So we'll give a radiation source. It'll start causing things. Uses dyes that emit visible light, bombarded with shorter UV rays, fluorescence. So we can dye certain things with fluorescent dyes, and then when the ultraviolet light hits it, it glows. All right, so super cool. We don't have one here yet. Maybe. All right, useful in diagnosing infections. Scanning confocal microscopes use a laser beam of light to scan the specimen. So you look, that gives you a lot more 3D comprehension of it. Integrates images to allow focus on multiple depths or planes. So if you were in lab, we looked at some of the plants and some of the Spirogyra and things like that. And a couple of you made a comment and I made an announcement. If you use the fine focus back and forth, you're going to be going in. That is so large that you're going to be taking transverse almost like transverse plane images as you go up and down. So as you toggle back and forth with that fine focus, you're actually going to see different depths of that same picture. So you can think it's stupid, or you can think it's kind of cool that you could do that, right? Now, electron microscope, just realize this is what we are using now, or your textbook, a lot of books, or if you Google search something, it'll give you a scanning electron image of it. So it's super, super tiny. And if you look, magnification begins at 5,000, goes all the way up to 1 million. So we can look at viruses or super tiny nanoparticles with this. Forms an image with a beam of electrons that can be made to travel in a wave-like patterns when accelerated to high speeds. We won't get into quantum physics where is it a wavelength or a particle? And, they could say, well, with the slit lamp experiment, we could change that just by, do we want to look at it as a particle or a wavelength? It'll actually behave differently just by, the observer effect. So electron waves are 1,000 times shorter than waves of visible light. Remember I said the resolving power, the numerator, the smaller you could make that. For here, we're talking about 400 nanometers. Electron waves are 100,000 times smaller than that. So that numerator is even smaller. So now we know the resolving power is going to be greater. Electrons have tremendous power to resolve minute structures because resolving power is a function of the wavelength. All right, so if I asked you a question on resolving power, if wavelength is in there, it's probably going to be the answer. All right, so if we give you a different version of that same question, you'll know, what we're asking. Or it's just our way of asking you, does this student know how we determine resolving power? So if you knew that, you'd be able to ascertain or figure out what the answer was, contingent upon your now or being able to remember. So a lot of you can remember things, but we need you to actually use application and apply it. All right, sorry, sensors. So there's a scanning electron microscope. This will actually give you 3D view. So scanning electron microscope bombards the surface of a whole metal-coated specimen with electrons while scanning back and forth. It's kind of like sonar radar. It hits it and bounces back. All right, so specimen preparation for optical microscopes. We're not going to be doing any wet mounts or hanging drop mounts here. That would be if we were looking at pond water or something like that. Allows examination of characteristics of live cells, size, motility. So we could watch a paramecium move or something like that. Shape and arrangement. Fixed mounts are what we're going to do here. So fixed mounts are made by drying and heating a film of a specimen. The smear is stained using dyes for visualization of cells or cell parts. So give me 1 minute. I'm going to go right back. Sorry, I just want to grab this. So I think you might be able to see this. So if you look, see how cloudy that is? So this is literally, I put a drop of water on there, distilled water, and then I can take part of a little bit of from a swab or a loop, mix it on here, let it dry, and then we're going to heat fix that so that will, if it was potentially alive, we would, number one, would kill it, but we actually fix it to the slide so that when you go to rinse the slide after you've stained it, you don't power wash the microbe off of the slide. So a lot of you, if you don't heat fix it, and I did it myself. So once again, in lab, if you mess something up, don't get embarrassed, don't get all red, don't start sweating, because whatever mistake you made, I made it right over there in 1993 when I was in this very same room with Dr. Rowe. So whatever mistake you make, I've already done it. Trust me. If there's a mistake to be made, I've done it, and I've done it here. So rookie mistakes, or you're not thinking. The cool thing about making a mistake is you make it once or twice, and then you never make it again. So it's just realize that this is all a part of learning. Okay. And like I tell everyone here, you make all the mistakes you want here in lab. You don't want to make the mistake out there after you get your license or doing something. That's a little more awkward. All right. So fixed mounts are mounted. We dry these, and then we heat fix it to the slide. The smear is stained using dyes to permit visualization of cell parts. So when I first started doing this, we would give students slides like this first day and have them look at it. And it really was an exercise in futility because we didn't have the new microscopes. And depending on how thick this was or how I made them or whatever, it was almost impossible to see the organism. And the point was, this is why we stain it. So we can do a simple stain, it'll give us just morphology or shape, or we can use a gram stain or a differential stain. It'll give us the shape and it'll tell us something about the cell wall structure, or give us a little bit more information on what's going on. All right, so staining. Dyes are used to create contrast by imparting color. So whether it's positive or negative, it's going to absorb it or repel it. If it's a negative stain, it's going to repel it. And then we can use that for like capsules or negative staining. We'll talk about that here in a minute. So basic dyes are cationic, positively charged chromophore. All right, so the positive charge of the stain, all right, remember, inside of a cell, you should know is slightly negatively charged, and that's done by the sodium potassium pumps. It's always going to be slightly negative on the inside. And then, I'm sure your instructor talked about polarization, repolarization. So we want to make sure it's slightly polarized. So the charge is going to be slightly different. We always want it to be a little bit inside of a cell is always slightly negative. The dyes are positively charged, so positives will attract to the negative. The stain will imbibe into the organism, stain it. So now when the light goes through, we have an outline or some kind of structure to give us an idea of what's going on. All right, so in a perfect world, the positive would go in, it would attach positive and negative come together. It's going to stain that area inside of the cell, bacterium, or if it's histology, it's going to stain inside of there. So we have some contrast. Positive staining surfaces of microbes are negatively charged and attract the basic dye. So that's how that works. So if we have a positive staining, we probably deduce we're going to have a negative stain, right? So acidic dyes are anionic. All right, so negatively charged chromophores. All right, so they're going to repel it. Right. So microbes repel the dye. Microbes are going to push it away. The dye stains the background rather than the organism. So we can stain around it. So if we're talking about like theoretically like a capsule stain or something that wouldn't, some of the capsules, they wouldn't absorb the dye anyway. Or if it's acid fast or microbacterium, it has that mycolic acid on it. We can't get it to absorb it. At least we can stain the outside of it. Right? Sort of like contrast dye if we were doing a barium swallow or something like that, right? So simple stains, one dye is used, reveals shape, size, and arrangement. So in lab next week, we're gonna have you do a simple stain. Pick anything you want, methylene blue, crystal violet, eosinophil, saffronin, malachite green, whatever you want to pick, and just stain it. So it'll at least give us a contrast, but you won't be able to tell whether it's gram positive or gram negative. All right, so the most important stain we use in microbiology still currently more or less is the gram stain. Hans Gram designed it, and he was able to, after thousands of tries, he was able to figure out a way to differentiate between a gram positive and gram negative bacterium. And that was really helpful for us to figure out antimicrobial selection for organisms. So use a primary stain, crystal violet, and a counter stain, which would be saffron, to distinguish cell types or parts. So differential stains, gram stains, the most prevalent, acid-fast stains, and endospore stains. So we're going to try to do that here with, I think we're going to use, I think we'll use Bacillus subtilis. So something that can form an endospore. And those are really, really tiny. We wouldn't be able to see them with a microscope, but if I stain, if I use a negative stain, I can stain the outside of it. We can tell what it is. So if we had a regular gram-positive rod, and then we have gram-positive rod that has an endospore, we could distinguish them. All right, so endospores are, we'll learn coming up, super hard to kill. The only thing harder to kill would be a prion. And I mentioned last video I did, if you're looking at Lysol, kills 99.9% of all microbes. These guys can't kill the endospores. All right, they're like I said before, there are some that they found that are 10,000 years old. You give them the right environment, and they come back to vegetative properties, which is fascinating. All right, Clostridium difficile, all right, C. diff is an endospore former. That's how it can live in your intestines after you've killed everything else off. Structural stains reveal certain cell parts, not revealed by conventional methods. Right, so capsule or flagellar stain. So we're going to try to, we'll do maybe a flagellar stain on Pseudomonas, maybe, and maybe a capsule stain on some Klebsiella. I'll see what works or what would be, yeah, we'll try it out. And sometimes it works, sometimes it doesn't. So it's science. You know, if the stars are lined up right, it's a full moon now, that weird moon. I don't know. It's, what is it? May 27, 2021. So you guys can look that up, but there's some kind of moon. But if the planets line up and everything's right, they work. Okay. So the videos are not working, but here from your book, textbook, simple stain is going to give you just contrast. It'll give you nothing more than morphology. Gram stain, gram positive stains purple with crystal violet. The gram negative uses the counter stain saffronin to stain pink. Here is India ink stain. So India ink, that should stain the outside of it. All right. Light goes through the middle. All right. Acid fast stain. Here's flagellar stain of Proteus vulgaris. I just grew some Proteus. Yeah, vulgaris morbilis. Yeah, so hopefully that will be viable for our flagellar stain, and then spore staining, which is more difficult. So we'll try to find some kind of a spore former. We'll do that. Probably bacillus. Yeah, we're not going to get into the Clostridiums. All right, so the 6 eyes of culturing microbes, and I said this in the beginning. So we want to inoculate something, and you did your plates, or we want to find an inoculum, something you're swabbing, could be an an abscess or something like that. Introduction of a sample into a container of media to produce a culture or observable growth. All right, and I think I've got some E. coli that I got that actually has some cultures, and you'll see. And if you were in lab Tuesday, I pulled some of the plates out and I asked you guys to describe the colonial morphology, and you were able to tell, well, that's raised, that's circular, that was glistening, that was more matte, and we kind of played around with that for a couple of minutes. Isolation, separating one species from another. So when you do your streak plates, you do them properly, they should have different colonial morphology, and that colony came from one single bacterium that we were able to isolate through the streak plate method, and through binary fission, it made enough of them to make a a pure culture, an exit culture, right? Incubation under conditions that allow growth. So we're going to incubate around 37 degrees. Some organisms grow a little better at cooler temperatures, some grow a little bit better at warmer temperatures, but for the microbes we're using here, 37 degrees works great. Then we want to inspect it. What can we ascertain, figure out? We want to gather the information, and then we should be able to tell what it is. So when we talk at the end of the course about the enterics, the microbiome, we'll be able to tell these enterics. They're going to usually all be gram-negative, very similar characteristics, but there are some biological tests that we can do that would let us know whether it was, a lactate fermenter, or Proteus has some very unique urease properties that can cause kidney stones and things like that. Because it's able to produce urease. And we'll do methyl red, Wilkes-Prower to talk about fermentation. We'll be doing a motility test where we'll stab some of these gel tubes, and then we'll see if things move perpendicular to the stab line. We'll say, well, that thing has flagella, it's propelling, it's moving from the stab line. And we'll go over all this. Yeah, we'll go into some citrate production, differentiation, differentiating Pseudomonas that grows on everything and anything. So isolation, an individual bacterial cell is separated from other cells and has space on a nutrient agar. It will grow in mounds of cells. A colony consists of one species. And I said, if you do your street plate right, you'll be able to isolate one single bacterium, and then if it's viable, it'll grow into a very specific colony. So next week, after we do our street plates, when you come back, you should have different colonies. There'll be different size, color, the height will be different, or the border will be striated, or it'll be different. It could be glistening. So we'll talk about color, shape, anything you can differentiate or contrast, compare the colonies. All right. So morphology generally means shape, but for that lab and for this course for lab, morphology, when we use it here in micro, it just means, it does, like I said, technically it means shape, but when we say, what's the morphology of that culture? Well, what color is it? What's the texture? What's the sheen? Whatever you can tell me that's different. I'm going to grab right now, I'm going to grab a EMB plate, which is differential for gram-negative, gram-positive bacteria won't grow on it. And the super cool thing is, if it is a lactose that can digest lactose as its carbon source, it will ferment it. And the plate, you'll see, is going to have a very, very cool green sheen to it. All right. Let me just see if I can be patient with you guys. So I'm gonna hit the light right. So if you can see, that has like a really cool, and it was actually even better Tuesday, because I couldn't get this E. coli to grow, which is really irritating because E. coli grows everywhere, but I couldn't. It was the slowest, most sluggish E. coli I've ever seen, but I got it to grow, and I proved that it was E. coli, a lactose fermenter anyway, by that sheen. So okay, so this is an EMB and a blood agar plate. It's divided in two. So this is differential for gram-negative and selective. I'm sorry, selective for gram-negative and differential. So one side of this is blood agar. The other side is an EMB, which I just showed you. So that's the color it would start out at, right? And as it grew, it changed. It's got that green sheen to it. So this originally was this site here, that color. Oh, I want to just see if I can get in 1 minute. Sorry, I don't have a production team. So I'm going to hit that. So if I hit that right, you should see tiny rays, things that almost look, I want to say braille, maybe. So that's a colony. So literally that grew from one single bacterium through binary fission every 20, 30 minutes. 2 became 4, 4 became 16, and so on. So those little dots are single colonies. Right. So isolation techniques. This is a streak plate. I think I embedded the video I made maybe 2 years ago, or a year ago. I think right when COVID hit, so 2020, I made a streak plate video. And we'll go over it again. I'll go over it before we do lab, but we really want to isolate that colony. All right, so we're going to start with something sterile. We're going to swab it here, sterilize it, cool it, swab it again, sterilize it, cool it. So each time we do this, it's really called a serial dilution. So that by the time we're all done, we do our final swab, we're hoping that one single bacterium drops off here as we dilute it, and then that single bacterium will turn into a colony. You can see here, this is our initial inoculum. We have a bunch of stuff going on, ticks, fleas, everything. So it's concomitant with a bunch of different things mixed. Then we sterilize it, and as we sterilize it and run past here, we're diluting it. And then as our final swipe, if we look, that's one different, that looks like possibly Klebsiella. It's really shiny, but Klebsiella should be more pink. All right, but we look, that should be Staph aureus because it's yellow, I can tell. All right, but we're going to see that these are all different colonial morphologies, so I know there's different organisms, and these are all pure cultures. So I could swab that. I could swab that, gram stain it, gram stain that, gram stain that, and I should be able to tell exactly what's here. I didn't make this plate, but I'm going to probably say there's 3 different organisms here. All right. Here's pore plate. These are all forms of just serial dilution. Right. So we're just kind of diluting that population off in the hopes that we get one single bacterium to land somewhere, and then that single bacterium will grow into a colony. So this is the same inoculant, and this is how it grows. All right. So I still would prefer to do a streak plate. Would just be my form of doing it. So if a single species is growing in the container, you have a pure culture, but if there's many, it's a mixed culture. Right. Check for contaminants, that would be anything that's unknown. So in lab, I'm going to go around and tell you firmly, cover your plate, cover your plate, cover your plate, don't touch anything, cover your plate. So what we don't want to do is have a mold spore or something drop down. You've got your plate open, you're talking, and it's moving around. We don't want any spores to land on your plate and grow and contaminate your plate. I mean, it's going to happen periodically, but we want to minimize that. All right. So here's ways to identify a microbe. All right. So cell or colonial morphology or staining characteristics. All right. So we can stain it, look at it, shape, color during stain, morphology, looking at it on the slide, or morphology when it grows on the plate with the culture. Right. DNA sequencing is probably the most accurate and quickest. And biochemical tests determine an organism's chemical and metabolic characteristics. That's what we're going to be doing when we do the enterics. So can it ferment lactose? If it ferments lactose, it's going to lower the pH. If there's phenol in there, the red's going to turn to yellow. Does it produce gas? All right, so fermentation produces gas. That's how, beer and sauerkraut and kombucha, they're producing carbon dioxide. And I have one here. Let's see this, hopefully. All right, in there's a little glass tube. All right, and this glass tube is open side down. So if I were to inoculate this with something that fermented lactose and produced gas, number one, this is going to turn yellow. It's going to lower the pH indicator. And the bubbles are going to form in here. It's going to raise this glass tube with the open end up. And then the gas will be at the top. It's like a buoy. It'll pull it up. We'll see a picture of that coming up here in just a second. Immunological testing, there's are strips. You just inoculate it, and whichever one has the antigen antibody reaction, you know what it is. Right? So media provides nutrients in the laboratory. And in lab, I talked to you guys and I brought the nutrient auger around and I said, literally, theoretically, the microorganisms who don't use the agar, which is a sort of like a seaweed derivative, it doesn't use that for a food source. It really is just providing a surface. So I said, jokingly, I said, it's just a little mattress for the organisms to sleep on and multiply on. All right. The nutrients would really be on the very, very top layer of that. And then once the organisms consume all the nutrients and they're living in their own waste, they end up dying, right? So media can be classified according to three properties. Physical state, is it liquid, semi-solid, or solid? All right, so I brought a couple here. So this one is, that's like a solid. This thing is a motility tube. So we would stab so we go in here and we'd stab. This is an inoculation. We go in and we'd stab it straight down, pull it back up. So it'd be a stab line, vertical line. And then we come back, and if there's anything growing perpendicular to it, we know that microbe had flagella and it was able to move. It was reproducing and it was moving away from the Y plane, going into the X plane. Or Z plane there, I guess, depending on. All right. And then here is liquid. This is nutrient broth. All right. This is a nutrient slant. All right, so this is nutrient auger. And rotate that, but it's kind of at a slant. So when you grow this, it has a surface to grow on. So it's going to be at an angle. So here is brain heart infused broth. So if we have something that doesn't really want to grow, it's really, really sluggish, give a little treat. We put it in the BHI broth. Very, very yummy. So there's a broth liquid. It's the same exact thing, but it's in an agar base. So it gives it a solid surface to grow on. So now I can visually look at this. I'll go grab one in a second and show you. I did some E. coli on there. So I don't know how good my camera is on this MacBook. But there's nothing growing on there. Right. Here is the clear broth. Give me 1 minute. Just had an afterthought. I'll be right back. Sorry, I have to go back. All right, so this was the initial broth. This is one I inoculated. Yeah, I did this one on the 24th. So in 3 days it grew like this. All right, so this is called turbidity. You look, it's darker. Started out like that, now it's darker. So I know something grew in there, was growing in there. So when I shine light through there, it's going to be denser than this. All right, the only disadvantage right now between these two is I don't know if these are alive or dead because I can't take their respiration and pulse, right? I just know something grew in there. So it isn't pure like this. It's not sterile like this. Not inoculated. This one, it's inoculated. I know there's condensation in there, right? But you'll see the white, that's E. coli growing in there. Now the advantage to this over a flat plate is condensation. If it drops on here, will disperse the culture and destroy it. With this, the condensation is on the glass. It doesn't really hit the organism, but maybe at the bottom. But I can visually see this. All right, so there are some advantages to that. Okay, so chemical composition. So I'm sorry, liquid, solid, semi-solid, or solid. When we do the enterics, I'll pull the tubes out and we'll show you a semi-solid. So it's kind of like Jello that hasn't quite hardened yet, semi-solid. So chemical composition, we have synthetic, chemically defined. All right, so that means I have the chemical recipe. I can remake that whenever I want. All right, I know the exact ingredients. Complex, there's something in it, but we don't know the exact chemical nature of it. All right, so this one is brain, heart infused. All right, so we don't know what in the brain and heart of the mammal that's in there that the organism needs. So we can't synthetically make it. We just know that most organisms find this really delicious and they love to grow in that. So functional type. So is it general purpose, like tryptic soy agar or nutrient agar? So most things will grow on that, all right? Or is it enriched? Is it brain heart infused? Or, like blood agar, TSA with a 5% sheep's blood. It's enriched. So things that are really fastidious or really picky, or high-end bacteria, they need very specific needs, and this will satisfy one of their needs. All right, differential. I'm sorry, selective. So selective, we have media that will grow. We could have media that reducing media that will pull all the oxygen out or would reduce the amount of oxygen if the microbe doesn't have a way of dealing with oxygen. Oxygen's toxic to us and the bacteria, but if we have a way of dealing with it, we can survive in it. Some organisms are anaerobic. They can't live in an oxygen environment. So these organisms like to live deep in tissues where the oxygen concentration is lower. Some of them, like E. coli, prefers oxygen, but it can change its metabolism to live in a less oxygen-rich environment. Or like C. diff, Clostridium difficile, likes the large intestine because the oxygen concentration there is very, very diminished. It wouldn't do well in a highly oxygenated environment. But anyway, selective is we can mannitol salt. It is selective for Staph aureus and Staph epididymis because of its salt concentration. All right, most things can't grow on it, but Staph aureus or Staph epi can live on it because they can tolerate slightly higher salt concentrations. It's also differential because Staph aureus, this, turned this pink media yellow. If you look on the edge here, maybe you can see a little bit of pink. All right, so it literally consumed all the mannitol alcohol sugar out of the plate. all right, and grew. All right, so this isn't going to live much longer, but that's Staph aureus. It's on mannitol, so it's selective and differential. Right, so same plate, stuff growing on there. That's Staph epididymis because Staph epididymis didn't consume the mannitol sugar. It didn't change the plate from pink to yellow. So I know this is Staph. Staph because it grew. All right, because only Staph will grow on mannitol salt generally. But it didn't consume the mannitol alcohol. So I know this is Staph epi. Selective differential. And remember the EMB was selective for gram negative, right? But it's, yeah, differential for lactose fermenters because if it ferments lactose, it changes to that really cool green sheen. So I know that whatever's growing on here is gram negative, right? Because this media will inhibit the growth of anything gram positive. And I know it ferments lactose because it has that green sheen to it. Same thing. This is EMB on one side, blood agar on the other. All right, so this would be selective and differential, both. Anaerobic would be a reducing media. All right, so it's going to really kind of, I don't want to say absorb, but it kind of pulls out any oxygen in the environment or some of it. Transport media just keeps things alive, going from one location to the other. All right, so it's very, very enriched, specifically for transporting sensitive cultures. You want to keep it alive so we can study it. All right. So liquid broth is not solidified. That's solidified. No movement. There's solidified slant tube. This is actually Simmons citrate. So this will tell us a little bit later on. It'll tell us about whether an organism can use carbon as a source. And this can turn beautiful blue in the presence of some organisms. Okay, so what's going on? So there is a liquid media. Other one was liquid. Same exact thing as solid and a slant. It's nutrient, liquid, semi-solid, and there is on an agar plate. Semi-solid contains a solidifying agent, usually agar. Right. Solid, firm surface for colony formation contains solidifying agent. It's liquefiable and non-liquefiable. And agar, we bring the solution up to boiling, we sterilize it, we can autoclave it, and then as it cools, we plate it, and then as it cools, it turns into these super cool plates. All right, but remember, the organism doesn't eat the agar. The agar is only there as a solidifying agent. Okay, so there's so the most common use is solidifying agent. Agar is the most common. I think it's a seaweed derivative, I'm pretty sure, unless they changed it. It is solid at room temperature. Room temperature, solid. Liquefies at boiling. Does not re-solidify until it cools to 42 degrees Celsius. All right, I've seen that question asked on labs, and I've seen it asked in tests. So just kind of think about that. And 42 degrees Celsius is higher than body temperature. Incubator is 37 degrees, so is your body temperature, roughly. You guys should probably know that because you probably had your temperature taken 1,000 times since in the past year. Provides framework to hold moisture and nutrients, not digestible for most microbes. Some will, but most of them don't. All right. Nutrient broth is a liquid medium containing beef extract and peptone. All right, so this regular nutrient broth, nutrient agar I just showed you, solid media containing beef extra, peptone, and agar. Same thing, it's solidified. So like I said, I can grow in here. All right, but I can't tell. I know something's growing because it changed the viscosity. We'll change the viscosity and the turgor of it. Okay. Same with this. But I don't know if it's alive. I don't even know if that's alive on there. Looks like it might be alive, but until I re-inoculate something and grow it, I don't know if it's still alive. All right, so I could, if I swab something out there and put it on a plate and put it in the incubator and nothing grew, I would say, well, either I did it wrong or that stuff was deader than dead. So synthetic contains pure or organic and inorganic compounds in the exact chemical formula. So synthetic means I can make it. I have the recipe. I look it up. If I follow the directions, like making a cake, I can make it. Complex or non-synthetic contains at least one ingredient that is not chemically definable. So, like I said, brain, heart, infused. I don't know specifically what chemical or what compound in the brain or heart ground up liquefied mixture they like, but I know that that organism likes it or needs it. General purpose media grows a broad range of microbes, usually non-synthetic. All right, so that would be nutrient agar or TSA, tryptic soy agar plates. Right. Enriched media contains complex organic substances such as blood, serum, hemoglobin, special growth factors required by very fastidious organisms. So like a blood agar, we have chocolate agar if you want to grow some Neisseria gonorrhea. Strep kind of likes, it likes a blood agar plate. So it likes a TSA plate, a little bit of blood. Then we put it in a little chamber and drop a little votive candle in there, pull all the oxygen out of the environment, and that strep is happier than happy. And I did some Tuesday, so I think next week I'll come back and I'll show you beta hemolysis on that plate, alpha beta hemolysis of that strep pyrogenes. I think I used on one, and then I literally swabbed my throat. So I work in healthcare, so I probably have some kind of strep back there, and staph and all that good stuff. All right. Oh, there it is right there. So county and zone of beta hemolysis. So this was a blood plate. Which is this side here. That's what it should look like, very similar to that color. But depending on what's growing, if it hemolyzes, hemolysis, so hemoglobin lysis. So if it lyses that red blood cell and consumes it, it will be clear. So when you're in the lab, we've got some halogen bulbs I put in the lamps here. You can just look and see if it's partial or fully alpha or beta hemolysis. So selective media, like I said, the mannitol salt, the EMB is selective for gram negative. It's differential depending on what happens with it. Some things can be just selective. Like a McConkey plate will only grow, shouldn't say only, generally will only grow gram negative, but it doesn't change it at all right? But an EMB is selective for gram negative, and if it turns that green sheen, it's differential for a lactose consumer that ferments lactose. Right? And mannitol salt agar is selective for staph, differential between Staph aureus and Staph epi. Remember Staph aureus means yellow. The colonies it'll grow golden yellow, and the plate will change from that pinkish color to that remember I showed you that, and it has a little bit of pink left, what started that color, but it consumed all that Staph aureus. All right, here's chrome agar. All right, I just ordered a different version of this. So this is super cool. You had like a UTI, and we could swab it on here. Depending on how it grew, it'll tell you what the organism is. Isn't that kind of cool? So E. coli will be pink, Klebsiella is blue, Proteus is that pinkish color. So they put chemicals in there, right? And as the organism reacts with the plate, it changes the color. So you could streak plate this out, and you'd be able to tell exactly what's going on. Okay. There's what I just showed you, the mannitol. So they swab this. So you divide it in two. This is Staph aureus because it changed the plate. Staph because it's mannitol growing here. Changed the plate from pink to yellow. This is Staph epididymis. Number one, this is colony is white. These are yellow. I can tell by the mannitol consumption what's going on. Reducing media, I said, this is going to have to do with the oxygen concentration. Contains a substance that absorbs oxygen or slows penetration of oxygen into the medium, used for growing anaerobic bacteria. So when we do the enterics, we're going to be stabbing from the top down, and then we'll see on the stab line, how is it growing? If it's growing really, really well at the top, I know it likes oxygen. If it kind of grows in here really well, it likes oxygen, but not a lot of it. If it's growing really, really well down here, I know that thing's anaerobic. It doesn't like oxygen. It's staying the hell away from it. Right? So there's different concentrations of it. Here's carbohydrate fermentation medium. So what I showed you here, I have a picture of this somewhere. Yeah, so this is phenol red fermentation test. See this. Same as that. Right? So it started out with this color, right? And as it ferments, changes the pH, will change the medium to this color. And there's, I said this is open-ended here. So as the gas forms, it's going to bubble up in here. So this thing should float around like a buoy if the gas bubbles are in there. The glass tube in there. All right, I'm going to end this now.